In this video, I'm going to go through some more microscope slides. For those who didn't see the first episode, this is a collection of slides mostly from the 1800s, with lots of bugs, some other animals, and a few other groups in there too. This time there are also some slightly different kinds of slides and microscopes to use as well. This small insect isn't labelled, but I think he looks very funky with his stripy legs. Under the microscope, it's a great sample, being very well intact and very clear to look at. You can see this sort of skirt or overhang to the carapace, which reminded me of the true bugs, aka the order of insects Hemiptera. Things like shield bugs, cicadas and aphids. Looking into these, I came across water bugs of the genus Microvelia, and I'm very happy that this one could at least be closely related to those. This guy has very obvious spiracle openings all over its body, spiracles being tubes that it uses to breathe directly through its exoskeleton via simple diffusion of gases, rather than having a powered respiratory system like we do. You can see the eyes very well here, and the detail and clarity on these spiracles is really great. This is a disc shell, a kind of diatom. Just two very tiny dots at the lowest magnification, but as I zoom in it starts to look very cool. Diatoms are single-celled algae that build a silica dioxide shell called a frostule around themselves. These frostules offer protection but also focus light for photosynthesis. As light goes through them, they produce structural coloration by diffraction, producing this kind of kaleidoscope of patterns and subtle colours as I vary the focus here. On this one, a label is scratched into the side and it's kind of hard to read, let alone film, but it says it's the head of a chalk moth. What stands out on this are obviously the big feathery antennae, which allow them to smell their environments. A lot of moths operate in the dark, and as you might expect, other senses than sight tend to be heightened, and therefore the sense organs tend to be bigger. In some moths, including this species, the females have unfeathered antennae, but males have these which they use to smell the pheromones of the females, helping them to find mates in the dark. Now, as soon as I zoom into these antennae, you can see that the individual branches are themselves covered in lots of tiny hair-like structures. These further increase the surface area that can come into contact with chemicals, therefore increasing the moth's sensitivity to smell. Now, there are going to be quite a few insects in this episode. Next is the head of a water boatman. Like the water bug we saw earlier, these are in the true bug family, and they skim across the surface of bodies of water. These are predators, and it has a long proboscis which it uses to eat the larvae of other insects in the water. They also have very good eyesight, and this slide is a great example of the compound eyes that insects have. These are really just two big eyes, but they're split into many separate hexagonal tubes called ommatidia, which feed light down towards the light-sensitive cells, which are the equivalent of your retina. So this one reminds me of the mosquito in amber on John Hammond's cane in Jurassic Park. This is of course another kind of mosquito, a female of a species found in northern Europe. As you might imagine with a mosquito, you can see this long proboscis that it uses to bite into the skin of a larger animal and feed on its blood. But you can see more features here like the abdomen, hooked legs for clinging onto their hosts, and another example of those compound eyes. There are also two structures in front of the eyes which look a bit like eyes themselves, but these are called palps, and therefore chemoreception, just like the moth's antennae we saw earlier. Next is the parasite of a pigeon. Pigeon lice or feather lice feed on the skin or feather debris of birds. They have long dagger-like bodies which help them to weave in between feathers, and they have these sturdy, strong-looking curled up appendages, and that will help them hang on to the bird when it's flying or bathing to try and get rid of things like these parasites. They'll spend their entire lives on birds from eggs to adults, sometimes transferring in between individuals, and that's how they spread. It seems a lot more complicated at the front end, and at the back is just this big abdomen. I guess that's kind of just like a big stomach to hold food. This one in the blue and gold slide is a parasite of a bat. This sort of looks like a flattened six-legged spider, but it's actually another fly like the mosquito, just in a group that has lost its wings. This is because its entire life cycle is spent on bats. so they have no need to fly around, and so they've lost their wings through natural selection. These sturdy legs with hooks and barbs help them to burrow through dense bat hair and stay attached to the animal. They also have barbs around their mouth, and of course these probably cause terrible itching for the bat. There are also some dark patches which at first I thought were eyes, but at higher magnification you can see that these are actually comb-like sense organs, and it turns out that these flies have also lost their eyes as well as their wings. They probably sense their way around with these hairs all over their body, so they really don't need to see. This is a male flea. You can see that these back legs are very long and almost look springy. Famously, these allow fleas to jump many times further than their own body size to find new hosts. Just like the bat fly, they have spiky barbs on their legs and mouth parts, all of which help them to attach but will cause irritation for the mammal they land on. Let's have a bit of a break from insects. This is a snail or gastropod. In life, snails of this species have stripy shells and live in the Indian and Eastern Pacific Ocean. From this cross-section you can see the cavity and internal structure of the shell. The snail grows its shell from the wider end, slowly secreting more shell in a spiral pattern around this central column. The outer shell has a few different layers. The innermost is very smooth and shiny, and the material is called mother of pearl or nacre. The outer layer is the hardest and is often coloured or patterned for display. You can see all of these lines perpendicular to the direction that the shell points in from base to end. This shows you the different layers that are put down slowly over time, one on top of the other, as the snail slowly grows. This one is a sample from a sponge, gathered in 1870 apparently, and from a polar region. For comparison, here's some footage of some dead sponge I found washed up on a shoreline, but this is unlikely to be the same species. Zoomed in you can see a skeletal matrix made of a collagen protein called spongin. Dotted throughout this are tiny spine-like structures called spicules. Some types of spicule you might have seen in the previous video, and these could either be for defense against predators or parasites. Sometimes they can also help strengthen the sponge's skeleton, or sometimes they might help attach it to a surface and keep the sponge in place. Next are the pedicellary or tube feet of a sand urchin, otherwise known as a heart urchin or sea potato. Sea urchins are echinoderms, just like sea stars, aka starfish. Echinoderms move themselves around with hundreds of these tube feet, which are these flexible extensions of something called their water vascular system. This system uses muscles to pump water throughout the body and move these tube feet via hydraulic pressure. This allows the sea urchin or starfish to move around hunting for food. Just one more insect and then I'll get onto some different kinds of microscope slide. This one is a female sawfly. These have larval stages which look a lot like caterpillars and eat leaves, but they grow up to look like this and become carnivorous, and you can see these pretty mean looking jaws used to do that. They're called sawflies because of this saw-like part on the back end of females called an ovipositor, which sort of means egg placer. They'll use this to cut into a plant to lay their eggs, and then the larvae will have a sheltered place and a source of food as soon as they hatch. Now, the slides we've seen so far have worked on the basis of shining light through the specimen into the lens of the microscope. But for specimens that are a bit too big, tough or opaque to be sliced into a very thin cross-section, you can use a slide that's basically more of a case and then zoom in on it from above. For this you use a different kind of microscope. Rather than shining a light from below, you use these kind of binocular lenses to look down on the subject from above. And it has this small lamp which you shine down to illuminate your subject. So let's have a look at these slides. First we have the elytron of a weevil. Now weevils are a kind of beetle with a long snout, and elytrons are the hard cover that goes over their front wings to protect them when they're not in use. This species is known as the gold dust beetle, and you can see why from this glittery pattern. This species is found in Southeast Asia, and they're often seen as pests on the leaves of agricultural crops. Obviously not sure what this one says, apart from being some sort of shell. You can see all these layers with struts in between them for support. This is probably from a marine organism, and the shell will primarily be made of a form of calcium carbonate. This one says parts of diamond beetles, presumably parts of the outer exoskeleton, similar to the weevil wing covers we saw earlier. That's it for now. Thank you very much for watching. I have footage of a few more slides, but the video was already quite insect heavy. Maybe I'll make some short videos with those, so stay tuned for that. Thank you again for watching.